So one of the difficult things about talking about this chapter is it feels so general and so vague. We've said that this activates a bunch of downstream targets or something like that, right? I understand that that's general and that's vague. This is the reality. This is the reality of signaling. And so we can identify parts here that look similar. Here's a G protein coupled receptor. Okay. Here's adenylyl cyclase. Here's cyclic AMP. Here's protein kinase A. What I want you to take from this, right, from this pKa signaling, is look at the number of arrows that are coming out of pKa. Look at all of the different targets that it has. So it's difficult, right, unless you're in a class that studies protein kinase A, right? I can't imagine that class, to go through all of the different effects, right, that pKa is going to have. And it's going to be true for the kinases that we talk about as well, besides pKa. So I just want to give you a sense of the incredible diversity of pKa signaling, right? Most roads are leading to activation of protein kinase A. You can see it right in the middle here. But I don't know, just the sheer number of effects that it has within the cell are huge. They're huge. Okay, let's go back to some of the other kinases and how they're regulated and some of their targets. All right, number 2 in our protein kinase discussion, something called calcium calmodulin dependent kinase 2. This is a mouthful, right? This is always abbreviated. Nobody wants to write that out. CAMK2. So this will be the way that I think this refers to it in the book. It's the way that I'll refer to it. CAM kinase 2 has 14 subunits. It's a big protein. Each individual subunit has its own catalytic and regulatory domain. So 14 subunits, each with a catalytic and a regulatory domain. Now, the way that this kinase is activated is, remember we talked about how calcium can come into the cell and bind up calmodulin. And so now we get this complex of CAM, that's calcium bound to the protein calmodulin when it comes into cells. CAM is able to displace the inhibitory domain from the catalytic site. Okay, so this protein actually folds back on itself and is what we call autoinhibitory. CAM displaces the autoinhibition, right? The inhibitory domain that's associated with the catalytic site. And oftentimes a target of CAM kinase 2 are ion channels. Okay, so CAM kinase 2 tends to phosphorylate or phosphorylate, not dephosphorylate, ion channels. So CAMK2 now active. Again, it has many, many targets. I could show you a figure just that looked like pKa for CAMK2, but one common target that becomes important for us is ion channels. So CAM kinase 2 phosphorylates multiple targets, including ion channels. So let's take a look at CAM kinase 2, right? It's letter B here. So you can see what I mean about autoinhibition, that the catalytic domain is hidden, right? So here's the catalytic domain here, and because it's bound to the regulatory domain, we think of this as being off, okay? CAM kinase 2 can come in and there's a binding site on the regulatory domain. It's small, but it's, I'll write you see this little dent here, pictured as a dent, right? So calcium calmodulin complex, this thing, can fit into that space, causing a conformational change. So now we relieve the inhibition, and now the catalytic domain is able to phosphorylate downstream targets, right? So by, you can see here binding the regulatory domain, CAMK2 is now active, and each of the 14 subunits are going to work similarly to this. Okay. Next, protein kinase C. Okay, the third protein kinase that's regulated by second messengers is PKC, protein kinase C. So if you remember, think back to the IP3 DAG second messenger pathway. Right? Once DAG exists in the membrane, right, that triggers the translocation of inactive protein kinase C to the membrane. So remember, PIP2 gets cut. That's my scissors. It gets cleaved into DAG and IP3. The presence of DAG, right? So with DAG being present and stuck in the cell membrane, that triggers this inactive kinase PKC, right, which is somewhere within the cell, and it's off, right? It's not active, to translocate to the cell membrane where we find DAG, okay, where we find the diacylglycerol. When it does this, right, it binds to the DAG, but also calcium and another membrane phospholipid. So PKC binds the DAG. It's a calcium-dependent process, so calcium has to be present here. There's a second membrane phospholipid that has to be present. So we'll just write PL for phospholipid. And when inactive PKC binds to the membrane and binds to all of these things in the membrane, at least the DAG and the phospholipid, these are in the membrane, and the calcium is also needs to be present, right? This activates, or we should say relieves, that same autoinhibition, and now PKC can be active. So we get active PKC. Okay, so again, I think maybe the picture is most helpful, right? We have a protein here, PKC, that's autoinhibited. Its regulatory domain is bound to its catalytic domain, right? So let me maybe write what these are here. So, oops. This is the catalytic domain, and this is the regulatory domain. Now, when this thing associates with the DAG, those are these little triangles here, as well as some other phospholipids here and calcium that's required, so it's a calcium-dependent process. Now, this kinase is active. So now, this PKC can leave and target other intracellular proteins and things of that nature, okay? So it has to basically go and get some activation at the membrane, has to become active at the cell membrane before it can go back into the cell. So these other two, right, PKA and CAM kinase 2, both of these are going to be activated within the cytosol. They don't need to go to the cell membrane, and they're going to have targets that could be in the cytosol. The targets could be in the membrane. Sometimes the targets could even be in the nucleus, right? We saw the diversity of targets. And the same is true with PKC with respect to a target. Wide variety of targets. Okay. Next on our list of kinases are the receptor tyrosine kinases that I talked about, right? And so notice I've jumped back a few slides because this has the best picture of what we'll use as an image of a receptor tyrosine kinase. So RTKs. So the first three kinases that we mentioned all phosphorylate serine or threonine residues of targets, okay, of target proteins. Receptor tyrosine kinases obviously don't, right? They're the ones that are going to phosphorylate or transfer phosphate groups to tyrosine residues, right? That's what phosphorylation is. So they have an extracellular domain where ligands bind. those ligands could be growth factors, could be various cytokines, typically not neurotransmitters, but other signaling molecules, okay, that affect changes, substantive changes within neurons, okay? So the ligands for this, I don't think of the ligands typically as neurotransmitters per se. You know, it's not going to be dopamine or GABA. Things like growth factors, which may change the genetic programming of the cell, or cytokines, right? Things that regulate, I think of cytokines as regulating inflammation. These typically activate receptor tyrosine kinases. The catalytic domain is intracellular. And its targets must either move or be present at the cell membrane. Okay, so whatever it's going to phosphorylate, whatever protein target it has, must, if it's away, if it's in the cytoplasm, cytosol, must move to the active enzyme, which is in the membrane. So the targets of this protein must move to the membrane, right? So if it's going to phosphorylate, if it's going to be phosphorylated, it has to move to the membrane. And I showed this picture because we're talking about these enzyme linked receptors here, right? So here's a, I don't know, maybe it's a growth factor, right? This binds to the receptor, and now this kinase is active. So it takes an inactive protein and is able to phosphorylate it, right? So now this can go on to have these varying effects that I showed you, like with PKA, okay? Receptor tyrosine kinases, maybe one less step, right? Because the receptor is, or maybe multiple less steps, because the receptor is the kinase itself, okay? And can phosphorylate not serine threonine residues, but tyrosine residues of the target protein. The fifth and final type of kinase we'll talk about are called MAP kinases. Or mitogen activated kinases. They were first identified in cell cycles, so that's where the name mitogen activated comes from. So MAP kinases. So we've talked about how many protein kinases get activated by second messengers, right? So for example, PKA is activated by a second messenger. But sometimes kinases are activated by other kinases. But some kinases, ooh, that's ugly, but that says kinases, activated by other kinases. And that's what MAP kinases are. Kinases that activate more kinases. So I think that I like this example figure that I didn't pull from the book because it shows a pathway, an intracellular signaling pathway that deals with things that we've talked about, right? So here's our receptor tyrosine kinase. The ligand is bound here, right? And what you can see is eventually, right, we're not going to talk about every pathway, but we talked about RAS, right? RAS is a type of G protein. So this G protein is active, and what it does is it activates MAP kinase, or MAP3K, okay? MAP3K phosphorylates, activates MAP kinase, MAP2K. MAP2K activates MAP kinase, and now there are targets within the cell, Okay, so I'm going to wrap this up here. We'll start with this in the next video, talking through this again, but you can see that an inactive kinase gets activated by a different kinase. That's the idea. Okay.